for coming back. To those of you who came back. It's a little bit smaller this week, but that's good. So people who are really interested in chromosomes now. So I think maybe starting next week, I'll start recording who's coming to the class. And I think the 27th may be the last day to change. Is that right? You guys should probably know better than me. I think by the 27th, I'll have an official list of the class. All right, so last week I just kind of went over a big overview of a lot of different areas that this class is going to go over. And I talked a bit about human chromosomes and chromosome disorders and how we know how many chromosomes we have. And from now on, we're going to go into a little more detail about chromosome structure. And so it's useful that we're all on the same page and we know about the structure of DNA itself, which is pretty much the most important molecule in our bodies. And it's the basis of chromosome function. So if we don't understand DNA, we won't be able to understand how chromosomes work. So we're going to start. Let's see. Oh no. Okay, that's all it is. All right. I hope you guys can see. You don't need to read these small labels. The point is really that to get chromosomes in the end requires many, many, many steps, starting with the basic DNA molecule. And many different proteins bind to the DNA, and those proteins get modified by other proteins, and the DNA folds and twists and gets knotted up in a very complicated pattern until they finally make these familiar chromosome shapes. And I'll tell you at the beginning that it's still a very active area of research. So the DNA double helix was discovered back in the 1950s, maybe 60 years ago or so, and we've had 60 years to learn about DNA. So we know quite a bit. We know in a lot of detail everything that happens up until about this point. So up to here we have very good descriptions, and from here on down to the chromosomes, it gets a little bit murky and less well understood. We still know a lot, but it's still this is an active area of research where there's controversies and unknown areas, but it's also probably the most exciting part. So the part that I'm talking about today, while we know a lot of detail, it may not seem as exciting, at least for people who are doing the research. So we have to start very, very basic to get to understand the rest of it. So we need to talk about the physical properties of DNA itself. I think we all know pretty much that DNA is a double helix, and it's kind of twisty molecule, and it has AACG, T, and probably remember a lot from our high school biology about DNA. We're going to go over a little bit of its more physical properties. I hope this is new to some of you. So these are the properties we're going to go ahead and look at. So starting with just the basic shape and size of DNA. So we've heard that DNA is a double helix. What's a helix in the first place? So you probably know that a helix is a curve in 3 dimensions where you have varying x and y coordinate. This is x and y. And a constantly changing z coordinate. And DNA is a circular helix, the most kind of simplest kind, where the distance of this helix is always constant. So the radius of this line to this axis is always constant. And English speakers sometimes confuse the words helix and spiral. These are 2 words that are used somewhat interchangeably, but they're different. So helix is this 3-dimensional curve, and like a snail shell is a helix, and a screw thread is a helix. But a spiral is just a 2-dimensional curve like this. So if someone says DNA is a spiral, you can tell them, no, DNA does not look like this. This is a spiral, and this is a helix. So And helices are pretty interesting objects in their own right. They have a few properties. They've got, of course, this radius that I said is always constant. In DNA, the radius is about half of this, so 2.5 nanometers is the full diameter of the DNA helix. So this radius is roughly 1 nanometer. So remember last time I was talking about the size of chromosomes on the range of micrometers, a millionth of a meter or 1,000th of a millimeter. DNA is 1,000 times even smaller than that. So the basic dimensions of the DNA, of the fiber that makes up chromosomes, is 1,000 times smaller than the chromosome itself. Okay. So another property of helices that all helices have is their handedness. So any helix, in any three-dimensional helix, is either right-handed or left-handed. And that's an intrinsic property of the helix. It doesn't matter how you look at it, right? This can be confusing to people who are not familiar with it, but maybe the most common helix we encounter is a screw, or maybe in my generation it was a cord of a phone. I don't know if you guys still have this. Probably you're familiar with the phone cord that is this helix. And it only goes in one direction, and it doesn't matter which way you look at it. Obviously, the physical properties of the cord are not changing depending on how you look at it. So helix is either right-handed or left-handed. And I will show you how you can tell what that is. But for instance, this is another typical helix, this spring. It's either right-handed or left-handed. And the way you can tell is that if you point your thumb, and this is where the name comes from, if you point your thumb along the direction that the helix is going, if the helix curves in the direction of your right hand, the fingers of your right hand like this, then it's a right-handed helix. So that's just a two-dimensional picture. I should have actually brought a cord, but I hope maybe you can tell from the light that if you curve your fingers around in the direction of that spring, that it's going away from you like that. So this spring is the right-handed helix. So eventually you'll be able to just, after this class, you'll be in the habit every time you see a helix in nature or somewhere, you'll immediately be able to look at it and say, oh, it's right-handed or oh, it's left-handed. So this one is a right-handed helix. And it doesn't matter, of course, which way you look at it. And left-handed is the reverse. So instead of the fingers of your right hand, it's the fingers of your left hand. So if you take it doesn't matter which way you look at it. Even if you flip it upside down, then it will still be a right-handed helix. But if you were to look at it in a mirror, then it's reversing the chirality or the handedness of that helix. And so this mirror image of that spring is a left-handed helix. So as you go down this axis, for instance, the curvature is going in the direction of the fingers of your left hand. All right. So with this crash course in helix handedness identification, now there's a few helices up here that I would like somebody to volunteer to identify. So we already had a spring, so can anybody identify the handedness of this spring? We'll go with doing their hands. That's good. I'm going to call on somebody at random. Can you tell me which way it's going, left or right? No. Okay, so all right. Maybe it's too soon for that. Maybe I can get a volunteer. Someone's got to figure it out by now. Yeah.
Right-handed.
Yeah. Yes, just like the other spring, right-handed. I don't think all springs are right-handed, but yeah, you can see. And another way of kind of seeing that is that these things, the spring coils that are in front are kind of crossing they're going from bottom right to top left, and that's also something that right-handed helix should have in common. Okay, what about this vine? Did I already looked at it?
Left-handed?
Yeah, that's definitely left-handed. Yeah, because this part is pretty easy to see. You can see that around that way. A lot of these vines have a preference. So one species of vine might have left-handed helices, and one species might have right-handed, and some have both. But now we get to the actual important one. So what is this DNA molecule? Yeah. Yes, and that is a fact, and that's true of all the DNA in our body. So DNA is not just a double helix; it is a right-handed double helix. Okay. And it's always right-handed. All the DNA in our body, with maybe very few exceptions. Maybe sometimes it can just flip the other way for a brief moment, but it always comes back. It's always right-handed. So in other words, let's not make mistakes that these other institutes have made. And a lot of people will draw DNA. Maybe they get an artist to draw DNA for them, and they don't really know that DNA is a right-handed double helix. And so you get places like some Wikipedia places will draw DNA the wrong way. This is left-handed, clearly. Biology, which is actually a really nice place if you're interested in learning about anything biological. I'll write that down. So, they have a lot of online courses, including some on chromosome biology and microscopy, and they're done by experts in the field. So if you ever are interested in some topic, you can go there. But unfortunately, these guys have the wrong kind of DNA. And even the Nobel, so right here, this is money in Argentina. I guess they're commemorating science, but they didn't do it right because this is a left-handed DNA double helix. And this is from the Nobel Prize's own website, so even the Nobel Prize organization doesn't get it right all the time. But I think people complained to iBiology, and so they updated their logo and they fixed the DNA double helix. Now, especially whenever you see a picture of DNA, you'll be able to look and see if the artist got DNA right, or if they accidentally flipped the image. Okay, so what another property, we're just kind of working our way down the list of DNA properties. Another important property of DNA is it's called anti-parallelism. And that means that so the DNA is two helices that are wrapped around each other, and they're both right-handed. But even though they're both right-handed, they're going in opposite directions. So if you say the helix is going in opposite direction, that implies that there is some kind of direction to it. And even a single strand of DNA has some sort of directionality to it. It doesn't have a right end and a left end, but it has what's called a 5' end and a 3' end. And DNA, it can either be in a 5 to 3 orientation or a 3 to 5 orientation. And I'll explain what I mean by that. So that's useful because DNA is right-handed, but it can have two different orientations, and that does depend on how you look at it. So if you look at it this way, you can say it's going from 5' on the left to 3' on the right, and if you flip it around, it will have the opposite orientation. I say it has its bases oriented in opposite directions, so we have to learn what bases are. And we know the bases are A, C, T, and G, but it's a little more complicated than that. And there's a whole set of words that vocabulary that we use to describe bases and parts of these bases and parts of the DNA structure. So there are three things we can talk about when we talk about A, G, C, and T. We can refer to the base. We can refer to nucleoside, and we can refer to a nucleotide. These words are probably familiar to you. You've probably heard them, but a lot of people use these interchangeably, and even even working scientists sometimes don't recall the difference between nucleosides and nucleotides. So to get it straight, bases are just these differing molecules. So the thing that's different about every DNA base are these molecules here that are familiar: A, G, C, and T for adenine, guanine, cytosine, and thymine. In this class, we're not going to have to worry to memorize these structures. I've never actually been able to memorize them for more than was required to pass the next test. I'm not a biochemist, so you can always look them up. But so long as you know that they come in two different flavors. There are these big ones that have two rings that are called purines, and there's these smaller ones that have single rings, and those are called pyrimidines. So there's two kinds of bases. But the base just refers to this unique part of the molecule, but there's a lot more to the DNA, to the different steps in DNA. The next level up is called the nucleoside, and that's simply the base attached to this sugar molecule. So this is a sugar molecule right here. And again, we don't have to worry too much about it, other than there are 5 carbon atoms in this sugar molecule. And this particular sugar is called ribose, or it can be called deoxyribose. So if we have one of these bases attached here to the sugar molecule, then we have a nucleoside. But a nucleoside by itself cannot become part of DNA. It has to have one more thing attached to it, which is this bridging molecule called a phosphate molecule. Phosphate has this P in the center, that's phosphorus atom. Phosphorus makes this complex with some oxygen atoms, and it makes this negatively charged bridge that can either have more phosphates attached, so then you can have nucleoside monophosphate with 1 phosphate, or di with 2, or tri with 3. So you've probably heard of ATP, which is adenosine triphosphate. So that would be adenine plus ribose plus 3 of these phosphate molecules. That's ATP, and that is a nucleotide, not a nucleoside. So as soon as you get these phosphate molecules on, now what you have is a nucleotide. So a base plus a sugar plus a phosphate makes up a nucleotide. Okay. But confusingly, when we talk about DNA, we often will just talk about the bases in the DNA, because all this stuff is in common to every single one of these. So we don't really care too much about the different ribose molecules or the different phosphates, because they're all the same. So we might say this DNA is 100 bases long. What we mean is that there are 100 nucleotides in a row. Okay, we just talked about the bases. When I was saying the DNA had a particular orientation, and there's a 5' and a 3' direction that DNA can be in. That refers to this sugar molecule. You'll note that these carbon atoms are numbered, or not. All the atoms of the sugar molecule are numbered 1, 2, 3, 4, and 5. Sorry if that yellow is a little hard to read, but they all have a number here. You don't have to memorize all of them, but the 5 and the 3 are most important. And I think it's helpful to keep in mind that this 5', number 5 carbon, is not located on the ring of the sugar, but it's just one removed from this sugar molecule. Whereas the number 3 carbon is in this actual ring of atoms. So here's another look at that with the atoms blown up a little bit more, and you can see these are the bases that pair with each other, and then the sugar and the phosphate are kind of on the outside, making up this backbone of the DNA. And I think you'll probably know that the bases pair with each other and that's how the whole thing sticks together. And A and T stick together, and C and G stick together. So yeah, you probably are very well aware of that. Later we'll probably make use of the knowledge that A and T have 2 bonds connecting them, and C and G have 3 bonds connecting them. So C and G make a stronger bond together than A and T do. Okay, let's see. But right now we know enough to talk about the directionality and the anti-parallelism, because you can see that if we were kind of to unwind the helix and flatten it out in this diagram, we can see that one strand of DNA is connected to this phosphate group. Remember I said the number 5 carbon is off of the ring, so there's fifth position is off of the ring, and so it goes. So this is the number 5, and then there's a phosphate, and then it connects to the number 3, and then there's a number 5 again, and then phosphate and number 3. So the order here goes 5 phosphate 3, 5 phosphate 3. Whereas on the other strand, it's the opposite order. So if you were doing the same order, it would be phosphate 5, 3, phosphate 5, 3. Okay. So this connection, where the phosphate connects to the different sugars, is opposite on both strands. So even though you have 2 right-handed helices that are wound up around each other, they're actually going in opposite directions. And this directionality is actually important for the proteins that bind to the DNA, because some proteins will get on this DNA and want to go along the DNA in one direction. So this kind of protein might bind here, a protein may bind here and start to move this way, but if it bound on the other strand, it would move the other way. So anyway, that's what anti-parallelism means, that the 2 strands are going in opposite directions. So the next thing to understand about DNA at the physical level is the fact that if you look at this, a very accurate structure of DNA, you might notice that the gap between these 2 phosphate backbones is of different lengths. So if you look at the distance between here and here, that's much longer than the distance between here and here, and that's not a mistake, and that's not an accident. It's actually done on purpose, and it's has a very important biological role. But to understand where it comes from, it's just a consequence of the fact that if you took the DNA double helix and looked at it on end, so you looked at it from the top, these bases do not go right across the circle of DNA, but they're always off to one side. So if you have a pair of A and T binding to each other, they do not lie directly in the center of the circle of the DNA, but they're off to one side. And as a result of that, it directly follows that you have a major and minor groove, and hopefully this little movie can show you why that is. Let's see. Yeah. Okay. So you can see that if you change the position of where those bases are, let's say we line them up so that they are actually right in the same line, then the 2 grooves of the double helix will be exactly the same distance apart. But if you move it over, if you start moving the bases over, you can see that has the effect of wrapping one of the helical strands around the other one more tightly or less tightly. So just by changing the position of those bases, you can change the difference between the major and minor grooves, and the actual spacing is something like that. So that's not an accident. And another way of looking at that is if you just take a circle with 2 dots on it and roll it, and take a snapshot of this circle rolling, you will get the same kind of thing. So you have a major groove here and a minor groove there. And I should probably do I have the actual distance? If I don't have a slide of it, oh yeah, I do. Okay. So why this is important is that there are two ways for things to recognize DNA. It gives two options and two different interfaces to the DNA double helix that can be exploited by different molecules. So most proteins that bind DNA will bind to the major groove because if you look here, you can see that there's really a lot more. This is the major groove here. All the bases are really there and they're exposed. And the protein has a lot of information here. It can read all these bases, so it can look. A protein can bind to these bases very specifically by looking at the major groove. And in the minor groove, everything is very crowded and hidden, so proteins kind of see it all as just one blur. There are very few proteins that can actually bind minor groove, but mostly minor groove has been exploited by small molecules, very, very small molecules that can fit inside the minor groove. So we'll see examples of both proteins and small molecules that can get in there. This is a typical protein that binds to DNA called a transcription factor, whose job it is to find certain sequences on DNA and sit there. Whereas this is an example of a dye molecule that makes DNA fluorescent, and it sits in the minor groove. And due to the, I guess I didn't write the numbers down, but one entire turn is 34 angstroms, or 3.4 nanometers, and the major groove is twice as big as the minor groove. So the major groove is about 22 angstroms, and the minor groove is about 12. Okay, well, maybe not twice, but so major groove it should be 22 angstroms, and the minor groove is 12 angstroms. An angstrom is just 10 angstroms are 1 nanometer. Angstroms are even smaller than nanometers. And then the last physical property, I saved the easiest one for last. It's simply the pitch of a helix. And the pitch describes, for one complete turn, how much distance is traversed for the helix to make one complete turn. And of course, it's the same for both helices, or else they would get out of phase with each other. And in the case of DNA, one complete turn is about 10 bases, so every 10 bases, that makes one complete turn, and that is about 3.4 nanometers, or a little more than, it's a bit larger than the diameter of DNA. So this is showing pretty much one and a half complete turns of the DNA double helix. So I said that all of the DNA in your body is right-handed, and that's true. But in some cases, you can make DNA adopt different structures, and it's not that important, except in some organisms that dry up and lose all of their water. They can temporarily put their DNA into a different form that can maybe compact a little bit more. So there's aDNA and zDNA, which are kind of non-canonical forms of DNA. And if you look closely, you'll see that zDNA is actually a left-handed helix. So this one is going in front and then passing around to the back. So if you ever see zDNA, that's actually left-handed, but zDNA can pretty much only exist in a completely dried up form. So it's not a living molecule. It's very hard to recover it back to vDNA. So inside our cells, there's really only vDNA. But when people were first, Watson and Crick and Rosalind Franklin and Maurice Wilkins were figuring out the structures of DNA, a lot of times they had dry DNA to work with, and so they were looking at aDNA and zDNA very often as well. But inside our cells, it's almost all vDNA, so we don't have to worry about the other kind. So we've kind of gone over quickly about basic structural properties of DNA double helix. So now we need to zoom out a little bit and look at the problem. The problem of how do we fit DNA inside our cells? And we need to think a little bit about length scales again. So the problem is that DNA is very, very long and straight. I showed you, it looks like it's curving a bit here, but this is probably, this is dried up a little bit. It's still very straight, right? It's extremely long, and yet it has to get packaged into a very, very tiny space. So this is a picture of E. coli, very common bacteria. It's less than 1 micron long, which is much less than 1 micron, probably 200 nanometers or so. And yet, it has 1.5 millimeters of DNA inside it. So more than 1,000 times longer. So imagine you've got to eat a string that is 1 kilometer long. That's kind of what you're getting all the DNA inside of an E. coli is like. To get an even bigger kind of length scale appreciation of our own DNA. So I said last time, each of our cells, we have about 2 meters of DNA. So much, much, much more than a bacterium. But if we took all the DNA in all the cells of our entire body, it would fit into a soccer ball like that. So that's really, really much bigger than an E. coli. And then the question I'd like to raise and then answer is, how long would that be if you took all the DNA in the soccer ball and stretched that out? So in one cell, we would have 2 meters of DNA. So if you took all the DNA in your body, how long would it reach? So, well, I just hope the answer surprises you a little bit. So let's say we start putting all that DNA, stuffing it inside the soccer ball. Is it bigger than the Earth? Would it stretch? How many times would it stretch around the Earth, do you think? Well, yeah, the answer, of course, if we knew the number of cells in our body, the answer would be pretty clear. And we have about 100 billion, it's not really known the exact number, but on the order of 10 to 100 billion cells in our body. So all the DNA in our body would stretch about as far as the farthest spaceship that has ever traveled outside of our solar system. So you have enough DNA in your body. If you stretched it all out, it would go way past the outer planets and go out to the farthest reaches of our solar system. And yet all that DNA is nicely packed inside our bodies. So biology has solved a pretty impressive problem of packaging. The solution is, so how is it forced to get inside this really, really tightly packed configuration? And the answer is, I showed a slide earlier, that there are these levels of compaction. And so the very first level we have to deal with is something that at first you might think would have an opposite effect, because the first step is DNA is wound around these proteins. So you're adding more mass to something that you have to pack, right? So you might think, why don't you just make the DNA really small? But it turns out that this wrapping the DNA around these proteins has the bulk effect of making it possible to pack it all. So to understand how we got to this picture and how we know that this picture is correct, I have to go over some basic stuff about how we can study DNA. If this is a little too basic for you, just bear with me, and maybe we'll go through it really quickly. The main way that we can study DNA, kind of a basic way, is we can separate them by size, and we can see how big DNA molecules are with a technique called gel electrophoresis. And this works by the principle of the fact that DNA is very negatively charged. Remember, I showed you all these phosphate groups, all those P's with O's on them. that are very negatively charged. So every base, every pair of bases has 2 negative charges on it. So if you have 100 bases, you've got 200 negative charges. DNA is extremely a negative molecule. Right, so that's those phosphates. So if you were to put it in an electric field where you have a positive electrode on one end and a negative electrode on the other end, and you put DNA in this gel matrix that doesn't let it escape, but lets it move very slowly, you can make the DNA molecules migrate towards this positive electrode. And because these DNA molecules are finding their way through this gel, it's actually an agarose gel that most people use. The smaller molecules can travel faster because there's less stuff getting in their way. So they all experience an electric field that's proportional to the same electric field, so they receive a force that is proportional to how much charge they have. But because the small molecules can move through this gel much faster, they end up at a lower position on the gel. So you can put all the DNA at one spot and then apply the electric field, and then longer DNA will end up at a higher position than shorter DNA. So that's probably what this looks like. And how many have you guys seen pictures like this? Are you all familiar? No. Okay. I'm glad you're not all familiar with this. So this is what it looks like when you're finished with this. So this is the gel apparatus you use. So it's really like a piece of, it's like yokan or content sitting in a buffer. And this positive field is made by this red electrode that you can see going here to this plus. So this is where the positive charge is coming from. And the negative charge is up here at the top. And the DNA is put into this gel here at the top, and after a certain amount of time, the DNA is being pulled toward this positive end. And you can see that they end up at different positions, different heights on this gel. So we know this DNA molecule, whatever it is bigger than this one, and here we've got some that are intermediate in size. So the rule for looking at these gels, if you see pictures like this, is always that big ones are on the top and small ones are on the bottom. And it's pretty much a logarithmic scale. So what does that mean? It means that these are really small and these are really big, and it's all kind of spread out. Okay. So I said this is DNA, but it's obviously not one DNA molecule. It's millions and millions of DNA molecules that are sitting here at this position in the gel. So how are we visualizing this? DNA does not just glow by itself. So we have to be able to stain this DNA with a dye. Remember last week I mentioned a little bit about different dyes that were binding to different parts inside the cells, and we have hundreds of dyes that would bind to DNA. The one that's used most commonly is this. That's called ethidium bromide. So it's a bromine salt of this molecule called ethidium, and when it gets into DNA, it makes DNA glow. So it doesn't really glow outside of DNA, but when it binds to DNA, it makes it look like this. So if you actually looked inside, if you looked at the DNA, it doesn't actually make it glow. You need to shine UV light on it first, and then it makes it fluorescent. It doesn't just glow by itself. You have to add UV light to it. Curiously, if you look at this molecule, I hope it kind of reminds you of something. It kind of looks like DNA itself. It kind of looks like maybe an A binding to a T here. Maybe this is the A because it's got 2 rings here. The T has 1 ring there, and that's not a coincidence. In fact, ethidium bromide binds to DNA by actually going in between the bases. So it's called intercalating. So if you have a sequence of A, C, G, where you have this ethidium bromide, there will be A and then ethidium on top of that, and then C and then ethidium on top of that. So it actually, when you add ethidium bromide to DNA, not only does it make it fluorescent, but it actually makes DNA longer. So you have to make sure when you do this experiment, that you add DNA to this gel after you run it through the gel, or else you may be changing the lengths of the DNA. Okay, so this tool of being able to tell how big a piece of DNA is probably the most basic but also most useful tool in molecular biology. It's just a routine thing that we're doing every day. So in my lab, probably we do this gel experiment 10, 20 times every day, and sometimes more if you have a particularly high load of this. And the main reason we're doing this is just to check that the DNA we are using is the DNA we think it is. Very often you'll have some DNA and you'll just wonder, is this really the DNA I think it is? And you have to run it on this gel to make sure that it's the right size. And if it's exactly the right size, then it's very good. It's very likely that it is the right DNA as well. So how can we make this pattern? So, right, if we have a piece of DNA, it should have just one size. But there's a way to take DNA and make it smaller in a very reproducible manner, and that is cutting it with these protein enzymes. So DNA can be cut by by a class of enzymes called nucleases. Whenever you see ACE, that usually means it's a kind of enzyme that does something. So there's nuclease, there's protease, there's kinase, there's phosphatase. If it ends in ACE, which mostly in Japanese it becomes ase, like nuclease or protease, that means that it's an enzyme. Okay. So for instance, there's a particular nuclease enzyme that scans along DNA, and whenever it runs into this sequence of 4 letters, whenever it sees this sequence of 4 letters, it makes a cut in the DNA right here. So it cuts the phosphate backbone. And while one enzyme might cut here, the other enzyme is cutting right here. And as you can see, CTAG on this strand is mirrored perfectly by CTAG on the other strand. So these enzymes, these nucleases, will usually cut what's called a palindromic sequence, or a sequence that is the same forwards and backwards. So if we take this plasmid and cut it with this enzyme, it will cut in a certain number of places. Because the sequence is very specific, it will always cut in the exact same places. The particular enzyme we have that cuts that sequence is called BFA1, and that's named after the bacteria that the enzyme was found in. And if we know the sequence of this plasmid, or if we know the sequence of this DNA molecule, we can predict the size of the bands that will result when we do this DNA electrophoresis. And we know precisely how long down to the base pair each of these bands is going to be. So this is a circular piece of DNA, and if we cut it 5 times, that will lead to 5 bands of predictable sizes. So we cut DNA and then put it in this gel and see this pattern, and then that's how we know what we have. Okay, but if we go back to the structure of DNA, this wrapping around proteins that I was showing you, that was discovered when people asked the question, what if we cut DNA randomly? So this particular enzyme does not cut randomly. It cuts at a very particular sequence. But there are other nucleases that just cut anywhere. They don't care about the sequence. They will just cut DNA at any position. And people were also examining what happens when you cut DNA with those guys. So if you cut randomly in a big piece of DNA, if you cut it once, you will roughly divide it in half. And then the more you cut it, the smaller the pieces will get. And so if you just keep cutting and cutting and cutting, eventually you get this distribution of sizes that has a very predictable distribution. And the longer you cut, the longer you add this enzyme, the smaller the pieces will get over time. And that was known very well for DNA from bacteria and viruses, which was the first DNA that people studied. So this was very familiar to a lot of people. And it was kind of harder to isolate DNA from eukaryotic organisms like us, like mammals and plants and fungus. It was harder to isolate DNA in a very pure state. But people wondered if our DNA would show a very similar pattern. And people finally succeeded at doing this. They saw something different. Okay, they didn't see this kind of smooth distribution of DNA, but they saw that DNA made this ladder pattern. So specifically DNA from eukaryotes like us, mammals and plants and fungus, if you cut DNA randomly, it doesn't make just a smooth pattern, but it makes this kind of banding pattern. And people were pretty smart, and they figured out that what was happening was, even though this enzyme should be cutting DNA randomly, it actually wasn't cutting randomly. That there were regions, regular spaced regions in DNA that the enzyme would not cut. So if the enzymes were cutting here and here and here, and they could cut anywhere within this region, but if they were blocked from cutting here, this would give rise to this kind of pattern. Because, for instance, let's say in this case, you'll get a piece of DNA that's this small that might correspond to here. And in this case, since it's randomly, if it missed cutting here, then you'll get a piece of DNA that is a bit longer that will correspond to this one. And so on, right? So this ladder was very intriguing to people, and the model was that that DNA must be organized in some repetitive, regular fashion, such that there's a protected region followed by a region that can be cut, followed by another protected region, followed by another region that could be cut, and that mirrors exactly the kind of picture that I showed you earlier of what DNA looks like under an electron microscope. So this picture that people saw in electron micrographs agreed very well with this data from random cutting and led to the model of that DNA is wrapped in this structure that people refer to as nucleosomes. And what is a nucleosome, right? Yeah, so the model became that DNA gets wrapped around a certain thing that turned out to be a protein, and then was exposed for a little bit, and then was wrapped around another protein, and kept going and going and going like that. And in fact, all of our DNA in our nucleus is like that. So what are these things that DNA is wrapped around? They're called histone proteins, and there's 4 different major histone proteins that are shown here in 4 different colors. And there's another bit of nomenclature here that has to be made clear. So histones are these individual proteins. Like this yellow thing here is one protein. This red is one protein. There's 2 of each, and they kind of mirror each other. So the red one, there's one here and one here. So each one of those proteins is called a histone protein. But then nucleosome is this entire thing that I'm showing you here. So a nucleosome is a set of 8 histone proteins plus about 200 bases worth of DNA that's wrapped around it. The exact amount of DNA that's wrapped around this nucleosome can vary from species to species or different parts of the same chromosome. But it's roughly between 150 and 200 bases. And this is more detailed of how these histone molecules get assembled. So the histones have different names, and they're always H something. So the H stands for histone, and then it's usually H followed by a number followed by 1 or more letters. But the main histones that we have are H3, H4, H2A, and H2B. So 99% of our histones, of our nucleosomes, have these four histones inside them. You might wonder what happened to H1. Well, there's an H1 as well, but we'll get to it in a bit later. But these four make up the core particle of the nucleosome. So the main set of 8 proteins that DNA gets wrapped around are these 4 histone molecules. And they assemble, before they get wrapped by the DNA, they assemble together in these pairs. So histone H3 and H4 come together in a pair, and H2A and H2B come together in a pair. And then they come together a little bit more. So 2 of these come together into 4, and then all together they come together into this 8. And a little more detailed view can be shown here. So here, hope these colors are distinguishable to all of you, but here the DNA is in purple, and you can see that now the DNA, now you can see kind of how the packing works. So if it weren't for wrapping around this nucleosome, this DNA would be stretched out in 200 bases, which that means there's 20 turns, and each turn is 3 angstroms, 3 nanometers. So that would be about 60 nanometers worth of DNA. Whereas here, we know that the DNA double helix is only 2 nanometers, and it looks like there's maybe 1.5 double helices of length here. So this is about 5 nanometers total. So this is a packing ratio, a length contraction of about 10-fold. So you're already reducing the length of the DNA by a factor of 10 just by wrapping it around this nucleosome. So we know DNA is right-handed. Maybe just a little test to see if people are retaining this. What about the larger coil of DNA wrapping around the histone? What's the handedness of that one? DNA wrapping around the histone, the core particle.
Right.
I disagree. You said right again. Yeah. I think, yeah. So this picture makes it quite difficult to tell because it's not at all. It's way too contrasty. But I think it starts here and goes behind and comes around the back, and then here it's coming around the front again, and then goes behind again. So that means this gyration is going that way, and the DNA is curved that way. So it's left. So DNA is right-handed on its own, but it wraps around nucleosomes in a left-handed, larger coil. These guys in Australia made a pretty nice movie of how DNA compacts and goes around nucleosomes. So I think probably seeing this movie will give you a little better impression. So yeah, that's just DNA by itself, a DNA double helix. And you can see minor and major groups pretty well. And you can see that it's kind of bending a little bit. And DNA does bend just from thermal energy from time to time, but normally it's pretty straight. Yeah. And then something's going to happen. Right, okay. So this is the first histone molecule, a group of 4 histone proteins just bound to its place on the DNA. And when it binds, so the histones are very positively charged, and they interact with the negatively charged DNA in such a way that it just encourages the DNA to bend around it. So it's a lot of electric interactions are getting together. And I'll stop it right there because we've already kind of moved into a higher level packing of DNA that is not as well understood. So from this point on, I have to say, from about this point, once these things start coming together in a higher order assemblage, we're kind of in a zone of, from here on, we're kind of in a zone of, we don't really know for sure what's happening. But this is one model of what's happening. So these DNA that's wrapped around these histones get together in a more complicated spiral pattern, and then these keep twisting and twisting again and again. This definitely happens in some organisms, but it's not clear that this happens in all of them. So you can get an idea now of how the DNA is continually packed in a series of hierarchical helices, and probably they're of different handedness, but this allows all the DNA to remain accessible and and then there's chromosomes dividing inside a cell. So a lot of that is still speculative, and we're still working on figuring out from the nucleosome to the higher level. But but it's not entirely wrong. Something like that must be the way chromosomes are packaged. Okay, we're done. There we go. Okay. All right. So okay. Just a little bit more on this core particle, because I said 99% of our histones, of our nucleosomes, have these four, H2A, H2B, H3, and H4 in them. But actually, each of those histones also comes in many flavors, many varieties that are used for different purposes. So although, yes, all of our nucleosomes have H2A, but there are all these different kinds of H2A. That are in different places in our chromosome, probably having different functions. One of them is involved in DNA damage, for instance, and one of them is involved in opening up DNA that's closed. And then similarly with H3 and H4 and H2B. So they all have many, many different kinds. Last thing about nucleosomes is that there is H1, but it's not like the other H2, H3, and H4. H1 actually sits outside of this nucleosome, and it's really involved in the next level up. So it's not involved in DNA winding around the nucleosome, but it links one nucleosome to the next nucleosome. And one theory is that depending on how much H1 you have, if you have a lot of H1, that makes the nucleosomes stick to each other a lot more, and it makes the DNA compact and go into a more higher order package structure. Whereas if you remove H1, then it makes it very loose. And this is kind of the diagram here, that these nucleosomes can either be far apart from each other, in which case the DNA is considered to be very active, and it can be accessed by proteins very easily. And we think that when genes are turned on, they're in this kind of active state where the nucleosomes are separate from each other. Whereas if genes are turned off, for instance, we have our chromosomes contain all of our genes in all of our tissue types and all of our cell types. So we have genes that we need in the brain, but they're in cells in our liver, for instance, right? We don't need our brain genes inside our liver. So we turn them off by compacting the DNA of those genes very tightly. This is one way that those brain genes are kept off where they're not needed. But in the brain, of course, they will be in this kind of active state where those genes can be turned on. And likewise in the liver genes will be in this active form. And it's a really active area of biology now. So how does the liver know to put the liver genes in this state and not that state? And likewise, how does the brain know to open up the brain genes and close down the liver genes? One mechanism that these cells use to do that is to not just leave it at the level of complexity of DNA wrapped around histones, but actually they change the tails of the histones. So histones all have part of the protein that sticks out from the DNA. And in the movie, you could actually kind of see it. If I go back to the movie really quickly, try to. I can fast forward, so we have to go again. But I'll try to stop it where we can see the tails of the nucleosomes. So we'll see how this can be a mechanism. So you see these things sticking out here that are kind of flapping around. These are several histone proteins that are folding the DNA, and these are globular masses that are binding directly to the DNA. But you see here, there are these bits that are sticking out. When I play the movie again, you'll see they're kind of flapping around very quickly. They're very accessible. So even though the DNA is wrapped around them and is kind of compacted, these tails of the histones, you can see them all now kind of flapping around there. They're sticking out, and they're like little tags or little switches that the cell can grab onto and use to change how all this gets packaged. It's not shown in the movie, but all these tails are continually being modified with other molecules. Okay. And some examples. It's not really important to get all these right now. One of the main ways to modify it is just by putting an extra methyl group, so just an extra carbon with a couple hydrogens on it. Just putting a methyl group, so an extra little, just one extra carbon is enough to really change the function and compaction state of these histones. There's also another, I'm not showing it here, there's another kind of modification called acetylation, but I think we'll just stick with methylation here because it's maybe the most important. Yeah, right. Yeah, but anyway, it's extremely complicated, right? Because there's several different histones, and not only H2A, but there's all those different flavors of H2A. They have many different amino acids in their tails, and each of these amino acids can be modified, some of them modified in many different ways. So the number of possibilities of histones interacting with DNA it eventually just becomes just mind-bogglingly huge. And we're wondering, is it really so necessary? Because in the end, it looks like the only thing that's going on is either DNA is getting compact or not compact. And there doesn't seem to be any other really large-scale effect of these histone modifications. It's either turning things on or turning them off. So we're wondering why it has to be so complicated. And do we really have to study all of them? Or is there some theory that explains how they make DNA compact and uncompact? Yeah. So when we talk about chromatin being compact and uncompact, that's kind of a layperson's way of describing it. The terminology that I might use sometimes is talking about euchromatin and heterochromatin. Euchromatin is good chromatin, and heterochromatin means other chromatin, which is, I guess, the part that's not good. They didn't want to call it bad chromatin. And these words came about when people looked at electron micrographs like this, where they know the nucleus is full of nucleic acid, but then they see some of these patches that are darker and denser than the rest of it. And the idea is that these dark and dense patches represent places where the histones become methylated, and the DNA is more condensed, and the genes are turned off. And we'll get to it a little bit later in class, but one very interesting observation is that a lot of this heterochromatin, where genes are turned off, seems to be located at the edge of the nucleus. So all the very outer part, the outer shell of the nucleus, seems to have quite a bit of this DNA that is turned off. And so why do nuclei do that, and do all nuclei do that, is an interesting question that we'll get into later. So for now, I think we'll skip ahead. To the last part. Right. So the final complication that's important to know, we're going back to bases here, is that not only can these histones be modified, so these histones can get methylation, but also DNA itself can sometimes be modified. A lot of organisms modify their DNA in many strange ways, but to keep it simple, I'll just talk about how mammals like us do it. And we only do we only do a very limited subset of DNA modification, and it only involves our cytosine residues. So we don't touch any of our other DNA bases. We just leave them alone for the most part. But cytosine is a little bit special in that we can very often take our cytosine bases and turn them into methyl cytosine, where the difference is instead of just a hydrogen atom sitting here, there's this methyl carbon atom, some hydrogen bond sitting there. So methyl cytosine works exactly the same as cytosine for almost all of our functions. So wherever you have a cytosine, you can replace it with methyl cytosine, and the genes will work exactly the same way, and the DNA replication works exactly the same way. So it doesn't do much. However, it is a difference, and it does lead to a difference in the condensation state of DNA. I'll just note in passing that methyl cytosine is a little bit dangerous to have in our genome, and that is because a very common chemical reaction can take methyl cytosine and change it into thymine. In other words, this part of the molecule can change to oxygen by a special reaction that just involves water. So at some rate, the methyl cytosine in our cells is turning into thymine, and thymine is a different base of DNA. So in other words, we can go from cytosine, a C, via methyl cytosine to T. So our C's that are methyl cytosine can sometimes change into a T, and that can lead to a mutation. This is just like our DNA gets damaged. And so we try to avoid having methyl cytosine in our genes where it's important. If we need a gene, we don't want to have it methylated. So it's usually avoided, but we still do it in case we want to turn off our genes. What's this one? Right, so if we look at where our DNA has methyl cytosine versus non-methyl cytosine, it's also in this heterochromatin where DNA is very condensed. And it kind of makes sense because if we have DNA that we don't need, like brain genes inside our liver, it's perfectly fine to turn them off with this methylation. Because even if it does turn into a mutation, it won't matter because we're not going to use that gene in the liver. However, sometimes if that gene does get turned on, it can turn into, it can give you a bad gene product, and that can lead to cancer. So this control of DNA methylation like this actually has a lot to do with biology of cancer, and we'll have another class on that. But here it's just showing where there's methylated DNA by these bright spots here. And this is actually fluorescence of a histone molecule. So you can see wherever histone molecules are located is in this left part. And it's the same cells, just showing you two different fluorescent molecules in the same cells. And when they're overlaid, it's red and green, so it may be difficult to tell, but you can tell, for instance, that the methylated parts of the DNA are in these kind of little balls all over the nucleus. And when chromosomes are dividing, this is kind of what I'll leave you with here. You can see that it's really, as the chromosomes are getting pulled toward the daughter cells, the methylated DNA seems to be at the leading edge. So the chromosome is here, and it's being pulled away from the other chromosome as the cells are dividing. So this is just a kind of basic illustration of how cells divide. Pretty simple. But the methylated DNA seems to all be here. So why should that be? Well, we'll get to that in another class. So that's just an interesting observation for now. So the very last thing is that if DNA gets methylated, there should probably be a way to get rid of that DNA methylation because, like I said, it's dangerous. It can lead to mutations. And sometimes you may want to turn off genes for a while, but then turn them on again. So there should be a way to get rid of the methylation. But unfortunately, this methyl group is actually quite difficult to remove when it's sitting on top of another carbon atom. So this is a carbon here, and it's attached to another carbon here. And this C-C bond is very difficult to break, and actually no enzymes in our cells are able to break this bond. So instead of directly getting rid of this methyl group, what happens is these other enzymes called the TET enzymes, they start adding oxygen atoms onto this carbon. So they take our methyl cytosine, and you don't need to worry about the names here, but they keep adding oxygen atoms to this methyl group until you have this one here with 2 oxygen atoms, and this is a carboxyl group, and now this can finally be removed from DNA and turned back to the cytosine. So some enzymes can put on this methyl group to turn off a region of our DNA, and then there are other enzymes that remove the methyl group to turn it back on. It's a bit oversimplified, but right. The take-home message is that our DNA gets packaged into this structure with histones, and then by modifying the histones and modifying the DNA itself, it can be compressed to this heterochromatic state where it's really, really dense, or left open in a state where it can be more active. And so we looked at that a little bit last week when we talked about the banding patterns of a karyotype of the chromosome staining, and there's regions that are loose and tight, and that's why chromosomes get this banding pattern. So these tight regions, not only does the base composition differ, but here we would expect to find a lot more methylated DNA and a lot more methylated histones, and the structure is much more compact. And I'll leave you with this image. These are giant fly chromosomes. This is a tenth of a millimeter, so you can actually see these guys with your eyes if you looked on a slide very carefully. It would be very tiny, but you can't see the bands. But with a good magnifying glass, you can see this. So there's an example of alternating regions of euchromatin and heterochromatin. Okay, so I think we'll kind of wind up there. And so, like I said, we understand a lot about that process, the initial process of packing DNA around nucleosomes and how DNA gets modified. But to get from there to the actual chromosome structure is a bit less well understood. But there are definitely a lot more proteins that are involved in that process. We at least know the identity of a lot of these proteins. So I'll present what we know, and I'll try to make sure where it's to make it clear what's just theory and what's actually well known. Okay, so we have some time for questions as well. If you have any questions now or after people leave, you can come up, and I'm happy to talk about this.